Welcome to the Nervous System Part 3. Today we're going to look at what happens at a synapse, which is the gap between two neurons. As we begin, let's consider our essential knowledge statement, which says that animals have nervous systems that detect external and internal signals, transmit and integrate information, as well as produce responses. The last time we were together, we looked at the actual functioning of the axon in the process of sending this action potential, and we looked at each of the stages and discussed what happened. And you drew a diagram of those events. Remember, we started with number 1. Do you remember what the charge was at that point? Around a negative 70 millivolts. And at negative 70 millivolts, this neuron is not firing, it is not sending information. But as stimulus enters and sodium starts to come into this neuron, it gets closer to reaching this level called the threshold potential. If the threshold potential is met, then we get an all or none response. Remember, the neuron either fires or it doesn't fire. There's no partial firing. It either fires or it doesn't. And when the action potential is sent down the neuron, that charge difference is depolarized. And so the charge inside the cell actually rises due to the influx of sodium. So in order to return the neuron's internal electrical charge to negative 70, the sodium-potassium pumps are going to pump out sodium and bring in potassium to reestablish this charge. And they can get it to a negative 70, and oftentimes we'll see that they actually continue to pump until the value is even lower than that. And this ensures that the neuron is not going to fire and takes into account any minute amounts of diffusion that may occur that would still keep my neuron at a negative 70 millivolts so that it wouldn't accidentally fire at an inappropriate time. And so today we're going to look at what happens when an impulse reaches the end. So if we have our neuron, let's say we have a presynaptic neuron, and it is sending a signal downstream. The neuron ends with these little telodendria, and then it can talk to or communicate with a receiving neuron. So we'll call that the postsynaptic neuron. And today we're going to be looking at what happens in this area between two neurons. And that space is called a synapse. And the synapse has no direct contact. So we need some sort of chemical messenger to deliver the message from the presynaptic neuron to the post. And so this is a really classic example of how one cell talks to a second cell. And I want you to remember this as an example of cell-to-cell communication. And our primary communication today is going to occur through the use of a special chemical called a neurotransmitter. So when we look at all that action potential that we studied last time being generated along the axon, now the action potential reaches the end of the axon. How do we get the message from the presynaptic to the postsynaptic cell? That's what we're going to consider today. So that synapse, or that part, the space, that little gap between the neurons becomes really important, and that gap is going to be filled by, or the work is going to be done by, neurotransmitter. And neurotransmitters, we have several different kinds, and so here are some really classic examples. You'll probably want to settle on one or two that you try to remember their function. We'll look today at what acetylcholine does and what its specific example is as it works inside this little cleft in between a sending neuron and a receiving neuron. So we're going to consider those events today. There are some adaptations on the neuron that make it able to do its job. It has some very specific structural adaptations that allow it to perform this communication. And one is that in the membrane in this synaptic knob, there are calcium-gated channels that allow calcium to come in. So this portion of the neuron is very specialized. There are also sodium channels on the receiving axons dendrites. So these sodium channels are going to allow sodium to come in. So there's another structural adaptation. And then of course the whole membrane is fluid, and that is going to allow an event, a very specific example of exocytosis to occur. So you can see it right here in this area. So those three adaptations, the calcium channels, the sodium channels, and the ability to perform exocytosis, are structural features that allow a neuron to be very specialized with specific adaptations to do the job of cell-to-cell communication. So let's look at the individual stages of this action. So first of all, we have this nerve impulse that's arriving at the end of this axon, which terminates with these synaptic knobs. You see it's sort of knob shaped. And calcium is going to enter. Remember, along the neuron's axon up here, we had sodium coming in. But here, when we reach this region, the synaptic region, there's a modification in the membrane that it contains calcium channels that allows calcium to come in when the impulse gets here. So when calcium comes in, it interacts with these vesicles that are in the synapse, and notice they have this little content inside, and that is a specific chemical called a neurotransmitter. In the presence of calcium, these vesicles will fuse with the membrane, and that allows what was once inside the vesicle to be dumped out in this space, or the synaptic cleft. The contents, the neurotransmitters, are going to then bind with the sodium channel that's on the receiving side of this gap. And when they do, notice that if there's a neurotransmitter open or in place, the channel is open. When there's no neurotransmitter in place, the channel is closed. So the neurotransmitters actually open the ion channel, and when they do, sodium is going to enter into the postsynaptic membrane. And we've already looked at what happens when sodium comes into the cytoplasm of the neuron. We know that it's going to change the charge. And so that way the sending cell can talk to the receiving cell by allowing a neurotransmitter to come across this space or cleft and interact with the ion channel, open it up, and allow sodium to enter. So that's the basic way that most neurotransmitters are going to work. So in this cell-to-cell communication event, the action potential is going to come down and depolarize the membrane just like it did along the length of the axon. The difference is that this time calcium comes in, and that causes the vesicles to fuse. Remember, this is an example of exocytosis. And when that fusion occurs, the neurotransmitters are released into that synaptic cleft. And then the neurotransmitter binds with a receptor on the ion channel, opening that channel. And when the channel is open, then that's going to allow the sodium to rush in. So here's just another artist's rendering of the same event. So we have on this side of the diagram, we have the sending neuron. We can see the calcium is going to come in and cause these vesicles, the neurotransmitter containing vesicles, to then fuse with the membrane of this neuron. And when it does, then it's going to release the neurotransmitter into that synaptic cleft. And so now the neurotransmitter, let's say in this case it's acetylcholine, the acetylcholine is going to bind with the embedded little integral proteins that are on the receiving or the postsynaptic neuron's membrane. So this space in between here is the synapse. And the synapse is where the neurotransmitter, that's where the neurotransmitter is going to be released. In this case, we were talking about acetylcholine. And that opens these sodium channels. The sodium channels then will allow sodium to enter, and that way we get transmission of the impulse eventually down the receiving neuron if enough sodium comes in to cause the concentration inside to be raised enough to generate an action potential. So I'm going to show you this old video clip that will describe that in even more detail.
Our brains are constantly processing information, which requires communication between billions of nerve cells. Signals are passed from a sending neuron to a receiving neuron at a junction called a synapse. An action potential in the sending neuron travels down the axon until it reaches a synaptic terminal. The narrow gap between the synaptic terminal and the receiving neuron is called the synaptic cleft. The synaptic terminal of a sending neuron contains numerous vesicles filled with neurotransmitters, chemicals that carry information across the synaptic cleft. When an action potential reaches the synaptic terminal, the vesicles fuse with the plasma membrane of the sending neuron, releasing neurotransmitters into the synaptic cleft. The neurotransmitters affect the receiving neuron, changing the distribution of charge across its membrane. Let's take a closer look. An action potential is propagated down an axon by the opening and closing of sodium and potassium channels. When an action potential arrives at the synaptic terminal, it causes the opening of calcium channels, shown in green. Calcium ions enter the synaptic terminal through the calcium channels. Calcium ions bind to the vesicles containing neurotransmitters. This causes the vesicles to fuse with the plasma membrane of the sending neuron, releasing neurotransmitters into the synaptic cleft. The neurotransmitters diffuse